Alright, I'd like to welcome you to this tutorial on dihybrid Punnett squares. If you need help with vocabulary that we learned back in monohybrid test cross video, please go back and visit that, such as monohybrids, homozygous, heterozygous, dominant, and recessive allele. Please take the time to go back and review the beginning of that video to help you out. So dihybrid cross. Here I've already created everything, and I'm just going to show you how to find the ratios and such forth. So dihybrid is two genes. So in this case, we're looking at the two genes of plant height, as we see here, dominant versus shortness, and then we're also looking at red flower and white flower, so flower color. All right. So I went ahead and created my key. So the gene here is height and flower color, and you can see tall is dominant to short is recessive. Red flower color is dominant to white is recessive. And then if we go down and read the next sentence, it says two plants, both heterozygous for both genes, are crossed. So because it just says they're both heterozygous for both genes, it would be written like so. All right, with a dominant allele, recessive allele for the first gene for height, and then a dominant and recessive allele for flower color. So then once you get the parents, you have to, what you'll do is the Punnett square. So you can see this Punnett square is a lot larger. So it's a 4 by 4, so it's about 16 squares. Okay. So what we'll do is I'm going to go ahead and set up one of the parents here. So we know the parent is heterozygous from the sentence. So it'll be written like so. All right. And the way you do this is you just use the FOIL method. So you have two genes. You have one gene on this side of the line, all right, so over here, and then you have another gene that's over here. Okay, so what we're going to do is simply FOIL our first allele to the first gene, which is going to give us this. Then we need to FOIL it one more time, which will give us this combination. Then we still have one more way to FOIL. We still have the next allele of that gene. So we take the letter and go here, and that will give us this combination. And then we have it one more time, then we FOIL it here, and that will give us this combination. Okay, and since both parents are exactly the same, down the side will also be exactly the same. Okay, so now the way you fill in this graph, the way you fill in this Punnett square, I'm sorry, is that you just take the letter. So I'm going to take this top letter and I'm going to bring it down. So the top T, and I'll bring it down. I'll bring the next T over. Right, and then I'll bring the R over. Okay, so I just brought the R down and the R over. And that's how I get a homozygous dominant for both genes. Now, you always write it in the way that you see the parent. So you see that the parent has height, all right, and then they have flower color. So you always write it in that form, no other way. So I filled in every corresponding square, all right? And now let's try to figure out the phenotype, okay? So let's look for tall and red, and I'll just highlight this in red, tall and red. So this is going to be tall and red, okay? This will be tall and red. 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 This square will not be tall and red. This square will be tall and red. This will be tall and red. This will be tall and red. Okay. And then if we just keep going through, this will be tall and red, and then that's it. So since it's out of 16 boxes, right? So tall and red is going to be out of 16 boxes. Okay. All right. So we just need to count it up. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So that would be 9/16. Alright, pen's not wanting to work for some reason. There we go. So 9/16 for tall and red. Now let's look for short and red, and I'll do that in blue. Okay, so short and red. All right, so we know that in this third column here, these are going to be tall, so we don't need those. But this will be short and red here. This plant will be short and red, and this plant will be short and red. So if we just count that up, that's 3. So we're going to have 3/16 for short and red. Right, and then we have tall and white, and I'll do that in orange. So here we're going to have tall and white. Okay, here we'll have tall and white, and here we'll have tall and white. And that's also going to be 3. And then we have 1 short and white, which is down here in the corner, and that'll be 1/16. Okay. And then if you just want to write this into a ratio, it'll be 9 to 3 to 3 to 1. Okay? So, you can also do that with the genotype, all right? So let me go ahead and erase this so that we can look at the genotype. So the genotype, we have all these different combinations that we can get down here listed at the bottom. So if we go through and just look for each one, we're going to have one of these. So let me get this in black ink. We'll have one of these, so 1 out of 16. And I'm just going to put the whole number. All right, so that would be that one there. All right, then we'd have, let's see here, a heterozygous with a homozygous dominant. So we just need to look. So we'll have one here, and I'll try to do this check mark up here in the corner. All right, so we have one there, one here, and looks like we're just going to have 2. All right, and then we're going to have one of these, which will be right here. Okay, and then a homozygous, so we have 1. We'll have 2. And looks like just 2 for that one. Okay. And then for this one, we'll have 1, 2, 3, 4. As you can tell, it just went right in diagonal like so. So we'll have 4. Okay, and then for this one here, we'll have 1, 2. Alright, so we'll have 2. And then this one here will have, looks like 1. This one here will have 1, 1. And this one will have 2. Okay. So again, if we just add this all up, so we have 4, 8, 12, 13, 14, 15, 16. So it'll come out to 16. Now, normally you wouldn't do the genotype unless the question was specifically asking for it. So when it comes time, really just focus on the phenotype here. That's the only one you really need to focus on when it comes to ratios. Okay. So here are a couple practice problems. Again, go back through this video if you need any help with any dihybrid problems or anything like that. But here are 2 problems that I would like you to try before class. Have a great day.